Good day, this is the Professor, CBMD PhD, and today's Wednesday, April 12th, 2023. It is 5:57 and 25 seconds, a.m. Eastern Daylight Time, and today's research, nanoplasmonic imaging. So let's hop to it here. Nanoplasmonic imaging reveals real-time protein secretion. Date April 11, 2023. Source: École Polytechnique. Researchers have used a nanoplasmonics approach to observe the real-time production of cell secretions, including proteins and antibodies, an advancement that could aid in the development of cancer treatments, vaccines, and other therapies. The researchers have developed a novel optical imaging approach that gives a four-dimensional view of cell secretions of both space and time. And by placing individual cells into microscopic wells in a nanostructured, gold-plated chip, and then inducing a phenomenon called plasmonic resonance on the chip surface, they are able to map secretions as they are being produced while observing cell shape and movement. The key aspect of the work is that it allows researchers to screen cells individually in high-throughput fashion, collective measurements of the average response that many cells do not reflect their heterogeneity. And in biology, everything is heterogeneous, from immune response to cancer cells. This is why cancer is so hard to treat. At the heart of the scientist method is a 1 centimeter squared nanoplasmonic chip composed of billions of tiny holes and hundreds of chambers for individual cells. The chip is made of a nanostructured gold substrate covered with a thin polymer mesh. Each chamber is filled with a cell medium to keep the cells alive and healthy during imaging. As cell secretions are the words of the cell, they spread out dynamically in time and space to connect with other cells. Their technology captures key heterogeneity in the terms of where and how far these words travel. The nanoplasmonic part comes in thanks to the light beam, which causes the gold electrons to oscillate. The nanostructure is engineered so that only certain wavelengths can penetrate it. When something like protein secretion occurs on the chip surface, to alter the light passing through, the secretion shifts. A complementary metal oxide, semiconductor image, sensor and LED translate to shift into intensity variations on the complementary metal oxide, semiconductor pixels. The beauty of the apparatus is that the nanoholes, distributed across the entire surface, transform every spot into a sensing element. This allows them to observe the spatial patterns of released proteins, irrespective of cell position. The method has allowed the scientists to get a glimpse of two essential cellular processes, cell division and cell death, in the study delicate antibody secreting human donor B cells. They saw the cell content released during two forms of cell death, apoptosis and necroptosis. In the latter, the content is released in an asymmetric burst, resulting in an image signature or fingerprint. This has never before been shown at the single cell level. And because the method bases cells in a nutritious cell medium, it does not require the toxic fluorescent labels used by other imaging technologies. The cells under study can easily be recovered. And this gives the method great potential for use in developing pharmaceutical drugs, vaccines, and other treatments. For example, to help our researchers understand how cells respond to different therapies at the individual level. As the amount and pattern of secretions produced by a cell are a proxy for determining their overall effectiveness, we can also imagine immunotherapy applications where you screen patient immune cells to identify those that are most effective and then create a colony of those cells. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.